Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einevold. While the purpose of most models explored by computational neuroscientists is to describe some aspect of neural activity, they do not always aim to account quantitatively for a particular set of experimental data. For example, the extremely influential Hopfield model aimed only to highlight qualitative features of associative memory. As Wolfram Gerstner half-jokingly said in podcast number 22 on 40 years with the Hopfield network, from a biological point of view, everything was wrong with this model. When I, in the late 1990s, asked John Hopfield about what he himself thought could be learned from his model, he said it might be a metaphor for how memory works. Likewise, the great success of the balanced neural networks discussed with Nicolas Brunel in podcast number 34 was the general insight that balanced excitatory and inhibitory synaptic inputs onto neurons in the network result in a random Poisson-like spike pattern typically observed in cortex. It was not aimed at explaining one particular experimental dataset. However, other models aim to account for particular experimental data. The most well-known and celebrated example is the Hodgkin-Huxley model for action potential generation and propagation in the squid giant axon. Here, the model parameters were fit using one type of electrophysiological experiments, and then the model was tested with great success against observations from another type of experiments. For example, while the experimentally observed propagation speed of the action potential down the axon was 21.2 meters per second, the model predicted a propagation speed of 18.8 meters per second. This quantitative deviation between model predictions and experiments of only about 10% or so is quite rare in biology. In their paper, Hodgkin and Huxley identified a particular combination of model parameters that gave this good fit, and that is now listed in many computational neuroscience textbooks. However, a general problem when fitting a model to experimental data is that there is not a unique solution. In practice, there are often many parameter combinations that provide equally good agreement between model predictions and experimental data. The set of such viable parameter combinations becomes especially large when your model contains many model parameters and available experimental data for model fitting is limited. This is often the case if you want to fit spiking network models to experimental data. In today's podcast, I talked to Richard Gao about how to deal with this problem. Richard just finished his postdoc in a group of Jakob Macke in Tübingen in Germany and has now started his own group at the Goethe University in Frankfurt. In a recent paper with the Macke group, he introduced the tool AutoMIND, automated model inference from neural dynamics, for fitting spiking neural network models to experimental data. This tool combines forward modeling of spiking network models with inverse modeling based on simulation-based inference developed earlier in the Macke group. With this fully trained tool, one feeds in experimental data and gets out parameter combinations that gives model predictions in agreement with these experimental data. And the tool does not only give out a single parameter combination compatible with the data, it gives out a whole ensemble of such parameter combinations. In the podcast, we first talk about the problem of non-unique or so-called degenerate solutions to the fitting problem. There's both a technical problem that AutoMIND addresses and a conceptual problem. Is degeneracy a sign only of a limitation of our model identification procedure? Or is it also a feature of biological systems that maybe makes them more robust? We then move on to talk about AutoMIND in more detail, as well as the results from applying this tool when fitting the spiking network models to experimental data from brain organoids and mouse brains. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast, where you also can watch the videos. I really appreciate all of you who are already Patreon supporters. Thanks a lot. I will be most grateful if more join in to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community on social media or otherwise. Okay, here is Richard. Yeah, welcome to the Theoretical Neuroscience Podcast, Richard Gao.
Thanks, Gauta. Happy to be here.
So, I mean, this podcast is essentially about how to fit mathematical models or computational models of neural systems to experimental data, and then in particular to how to fit values of the parameters in these models. And I mean, you've been working. I mean, you were the. And now you're. I mean, you're just. I think that I met you at the Bernstein meeting,
Bernstein, yeah.
right? And where you organized a nice workshop, and then you had just started, like I think, an assistant professor position
Exactly.
at Goethe University.
Yep.
Yeah. So, what kind of position is that?
So yeah, I just started as a W1, which is the German equivalent to an assistant professorship here at the Goethe University Frankfurt, and in the computer science and math faculty. So yeah,
so I guess you will because this podcast will mainly be about, I guess, work that you did as a postdoc.
Exactly.
Jakob Macke's lab in Tübingen, right? For like 21 to 25 or something. I just asked my favorite AI or large language model to give you your CV.
Yeah.
And then before that, you did a PhD in cognitive science at UCSD.
Yeah, with Brad Wojtek.
In the lab of Bradley Wojtek, right?
Yeah.
So I mean, what did you do with Bradley Wojtek?
So with Brad, I mostly worked on analysis of neurosignals and mostly ePhys, both human non-invasive like EEG, but also invasive stuff like ECOG and some animal LFP and a bit of spiking data as well. Mostly, yeah, like intracranial ePhys data.
So that was more data analysis.
Exactly, yeah. Data analysis with a bit of statistical modeling. So like fitting basically curves to time series or the frequency spectra.
So, I mean, when you sort of do this I mean, if you want to fit a model to experimental data, then in general you run the model for many sets of parameters, and then you sort of traditional way is at least that you jump around in parameter space trying to minimize the deviation. And I think if you come from physics, and then you sort of, your intuition is that you're looking for one unique solution, like the perfect like that. But that's not really the case here when these biological systems, is it?
No, it's not the case. And actually, very often you find zero solutions. So how this all kind of started for me was in the context of my PhD with Brad. So we have some data, and I was trying to fit spiking neural networks to this kind of data. Actually, like the Brunel type of networks who you had on a couple months ago, I think. And one of the problems actually is if you just take random parameter sets off that model. In that case, there was just two free parameters, right? But a lot of other ones that in his paper, he had tried different values that's constant. But anyways, there's a bunch of free parameters. If you try random combinations, you essentially never find any reasonable regime. So in these spiking networks, very often you get these huge synchronous activity or like jail cell rasters, or nothing happens. So that's like the first problem you face when you're trying to fit these models. If you're lucky to get a model that works, usually you run with that, and that's great. Or if you're working from a previous paper, you just take their parameter sets and maybe move around in the reasonable working regime. So that's how I got started doing this whole business of trying to fit spiking network models or just biophysical models in general to data. And it's a painful process, right? So, I mean, I'm sure you can relate to that.
Yeah.
So I had met Jakob at a summer school in Lisbon. This was in 2019, and he gave a very nice talk, an earlier version of simulation-based inference at the time, essentially a method to fit these types of biophysical models, either single neurons or spiking neural networks, using machine learning as a sort of helper in that process. And that work was driven mostly by Pedro Gonçalves and Michael Deisler and Jan-Matthijs Wikman. Anyways, he gave this really nice talk, and I was sold because I had this problem, right? I had this data set I wanted to fit models to, and so I had asked to do maybe like a research internship or something. Eventually, I ended up doing a 4-year postdoc with him. So that's how this all got started. But
Because,
back to your original question.
yeah.
Sorry, go ahead.
So then, exactly. So then it first started off maybe that it's hard to get one reasonable
Any
solution.
model.
Any reasonable model. But then after you get this, at least one reasonable set of parameters that makes the model fit your data sort of, well, reasonably or quite well, maybe, then that's then it's sort of natural to declare victory and
exactly, yeah. Especially if you're a poor grad student like I was, you would have spent maybe 2 to 4 weeks doing this, and I'm happy with the parameters, and I move along with my analysis that I want to do, right? Exactly.
Yeah. But that's sort of the thing, right? Because it's, I mean, so even if you get then this one set of parameters, then it turns out, as you show in this, well, as you discuss in this paper, we actually also did some work on this in on a much simpler model back in the days with some firing rate models, but that you have some model parameters which are stiff and some models parameters which are, you call, sloppy. So what are the what are stiff model parameters? What are sloppy model parameters?
Right. So once you have this one model that fits the data quite well, if you are brave enough, let's say, to explore more space in the parameter or more regimes in the parameter space, what you can see very often is if you move the value of some parameters, they don't actually impact the outcome of your simulation or your model output that much, right? Whereas for some parameters, and those are what you call sloppy parameters. And for some parameters, if you change it even a little bit, the outcome of the simulation changes drastically. And so you would call those stiff parameters or stiff dimensions of the parameter space.
Exactly. So but what sort of ends up being sloppy and stiff, that must depend on how rich your experiments are, wouldn't it? Or I mean, how if you have a very sort of like if you want to fit a lot of data, I mean, I just said the intuition, if you have a little sort of just a little bit of experimental data that is not too difficult to fit, whatever that means, then you end up with more sloppy model parameters than you would have if the experimental data you fit to were more sort of extensive.
Yeah. So there's a technical point here on underdeterminedness or underconstraining or constraintness of your parameters based on the available data. And in general, and also another point is how precise you require the fit to be, right? So if you imagine trying to reproduce every single spike in a recorded spike train by a model, first of all, you probably would never find a solution. And even if you do find one, there's likely not that much wiggle room to still have a different parameter set that captures exactly that observation, right? But if you allow your reproduction to be less faithful, let's say, right? Like if you wear more blurry goggles, then everything else kind of looks the same. Then more stuff looks the same. So exactly. And another aspect of what you mentioned is the availability of experimental data. So the more data you have that you're trying to fit, the fewer solutions presumably there will be.
So this sloppiness, I think often sometimes when you end up with this sort of like sloppiness, then you can, one interpretation or maybe like natural, whatever type one thinking is that something you could do better or that is sort
Right.
of it really is a unique solution. It's just that you haven't
Right.
found it, or a less sort of but sloppiness is maybe not it's not maybe not a bug in some sense. It's maybe a feature of biological systems.
Exactly. And now you're touching on this point of we're moving from a technical aspect of maybe
Mm,
you're not constraining your model enough to having found real sloppiness or degeneracy. Degeneracy is the
mm,
term that you call this by in the system, which was, I mean, here you have to really give credit or a shout out to, Eve Marder, Astrid Prince, who really kind of, I think, spearheaded this movement maybe in neuroscience, and then before that, Gerald Edelman, who had this pretty famous paper of degeneracy in biological systems, which is a concept that in real systems you have degrees of freedom that allows the system to behave robustly to, for example, perturbations or just noise or stochasticities in its components, such that the behavior or the function is still maintained.
mm, mm. Because it gives you robustness in case, say, if something bad happens to your, like one ion channel, say, then
Right.
another ion channel can sort of upregulate and compensate.
Exactly. And so in Eve Marder and Astrid Prince's original work, it was this stomach circuit in the crab or lobster, right? And
Mm,
their manipulation was temperature. So if you move the temperature a little bit, some circuits are more robust to that perturbation.
mm. So yeah, because I've heard people saying that, well, one way to think about aging is sort of that you start young with a very sort of big parameter space which are
Yeah.
compatible with life, and then
Right.
and ion channels die or whatever goes down, and in the end you sort of the
Less
smallest kind of
compatible with life.
life. Yeah. And then when the final parameter things happen, you just die. I don't know.
Yeah.
It's a way to think
It's
about
maybe
it.
a bit morbid, but
Yeah.
yeah, perhaps you can think about that
Oh,
in
that's
the
okay.
framework of less flexibility in the
Yeah.
system to perturbation, right?
Yeah.
Such that in the case of, I guess yeah, you're flexible, less flexible to perturbation in the system in general, so that when something does happen,
Mm.
you have catastrophic failures where other components cannot pick it up anymore.
Mm. Okay, so you have this. Say you have found this one solution, and well, you start with a model, a one solution that really fits the data well, and then you explore this structure of the parameter space, maybe around this good solution,
Yeah.
that it sort of tells you the sort of the sloppiness structure, which tells something about the size of the parameter space, which is compatible with that particular function. So
Hmm.
I guess you called it. I think in your paper you called it sloppiness structure, or maybe or something that
Invariant structure, yeah.
invariant. So the point is that, but why is it invariant? I mean, it could change as you develop. I guess that's what one thing you find is that this sloppiness structure that change might change over time
Yeah.
when you
So
develop, or
the word I ended up using, which we had a lot of discussions about with the co-authors, is invariant. It's kind of so degeneracy is the general concept that you have
mm,
wiggle room in the system, right?
mm,
And the thing I am calling invariance is the structure of the degeneracy. So degeneracy is the description of the thing, and then invariance is, or the structure of degeneracy is the noun for the invariance.
okay,
Um, so it's invariant with respect to something. And that something here, in the case of the networks that we study, is dynamics. So given a specific kind of dynamic that you're trying to reproduce, like, bursting at a specific rate or oscillation of a specific frequency, your degenerate parameter space is invariant with respect to that. So you can move around anywhere in that parameter space and your output doesn't change. So
mm,
that's what I mean by
mm.
invariant.
That's
But what
okay.
you're referring to is that structure then changes over
Mm,
time, especially if you're trying to constrain it on different kinds of brain recordings, such
mm,
as the one that you get across development, because your network changes over time.
mm. So I think you also use the word functional equivalence, meaning that all these solutions within this invariant
Right.
structure, sloppiness structure, has the same kind of function. So
Yeah.
and so I mean, but so your paper is, I think, it's about this tool, AutoMIND, which you use where the goal is to then not only find this one good solution, but to really to find the whole ensemble of like parameter combinations for and for a particular application to spiking neural networks,
Yeah.
right? When fitted to
Right.
data.
Right.
So so that is sort of what this AutoMIND tool does.
Yeah.
And so when you sort of course we're coming to the details of that soon, but I guess when you want to do this kind of approach, I mean, you have the first choice is sort of, well, you need to have some experimental data that you want to fit, that you determine is sort of like showing some interesting dynamics that you can use to fit the model or to test and develop a model. And then you have to decide, I think you call it the summary features of the data. So that's because when it's like a spike train, there are many things you can try to reproduce, right? But
Right.
you have to make some choices there.
Exactly. So here, the summary features is kind of like what I was referring to before. They're essentially blurry goggles that you look through to some level of acceptability to say, I want my simulations to look the same as my experimental data only with respect to the summary statistics or like this level of blurriness. And so you can say, I have this spike frame recording from 1,000 neurons over a minute. There's obviously hundreds of thousands of spikes. I don't want to reproduce every single of those spike timing exactly, right? Because we know that there's a lot of stochasticity in the system, et cetera, et cetera. So what is a meaningful aspect of this piece of recording that is that you would want to reproduce? And that changes depending on the person, depending on the experimental context, and depending on the system you're looking at, right? So you might want to use, for example, like the ISI distribution, or in my case, in one of the examples, it was the rate of these network bursts in vitro. And then in the other example, it was the full frequency spectrum of these in vivo recordings in the mouse. So it really depends on what you want to capture as an experimentalist or as a theoretician, if you have specific theories or hypotheses about that particular aspect of the network that you want to capture.
So, and then you have to make, okay, so then you have like then you have decided on what aspects of the data you want to reproduce in your model. And then you have to choose, of course, the model structure. In your case, which you're coming to, it's a collection of like 2,000 neurons, iterative fire type neurons. And then you have to sort of also decide what model parameters to optimize. That could also be, that's also a choice, right? That you could decide to sort of to only, well, to assume that you know some of them and then keep them fixed and just only look at the others. So I mean, you now, I mean, you in this paper, you chose a model structure to account for both spiking in these organoids, these small, like this stem cell derived little pieces of neural tissue, and also in vivo recordings in mice, right? And then you chose a particular model. You chose something called the clustered spiking network model. So what was the model like?
Yeah, so the cluster spiking network model, it's at the single neuron level, it uses this neuron model called adaptive exponential integrated fire neuron, which is very similar to a linear integrated fire neuron or a lift neuron, which is sort of a phenomenological neuron model, right? And there's excitatory ones that are inhibitory ones, and then there are some there's individual parameters. Sorry, there's parameters at the single neuron level that are already free. And then you put 2,000 of these neurons in a network to connect them in a particular way, and that's the spiking neural network. So, and there there are some choices that we made on what the connections look like. So specifically, the clusterness refer to the fact that the excitatory populations have clusters that within the clusters there's preferential or there's increased probability and strength of connection. And then across clusters, they're basically weaker and less likely to be connected. And this is built off of a long strand of work. Specifically, the numbers we use, I think, was from this paper from Brent Dwyer and Ashok Levin Kumar on cluster networks.
Okay.
And they've had multiple papers now on these sort of spatial balanced networks. So that was a choice. And then there are parameters at the connectivity level, so at the synapses. So for example, the time constants of the synapses, their strength, and then there was also some parameters on the background drive, so the Poisson rate that goes into these neurons. But
So
the higher level consideration, I guess, was we wanted a model that was general enough to recapitulate multiple types of dynamics and to have enough free parameters such that there's the flexibility to move around in this model space to get all kinds of activity out.
So yeah. So then you had like, I think you also have conductance-based synapses. That was also a choice you made. Yeah.
Yeah.
So okay, so you had this quite general, or well, I mean, at least there was not the simplest kind of neurons.
Right.
It's not leaky integrating fire. It was like this adaptive exponential integrated fire neurons, even though they were obviously not sort of they were sort of point neurons, and then you had these sparse connections, but these clusters of excitatory neurons, and then you decided to have you had 28 free parameters. So because so that's also a choice, right? I mean, I guess in AI, when you sort of optimize parameters there, you typically keep the neurons fixed. Neurons are fixed, and you optimize the connection parameters, right?
Right.
But here you do both.
Right. Exactly. So in AI models, typically people fit deep neural networks. When they say we fit deep neural networks, it really means we are optimizing the connection weight strength between layers or between the neurons in the network, right? And I think a very underappreciated effect of biological brains that hasn't really made its way into machine learning and AI is the fact that single neurons have very different properties, right? Even the same type of neurons, like a classical pyramidal excitatory cell. If you take 10 different pyramidal cells, they have very different intrinsic properties, like membrane capacitance, different expressions of ion channels, etc. So that was a conscious choice to say we allow those single neuron parameters to also vary, just to see what their effects would be on the total network dynamics.
Okay, and then you had made all these choices, and then you started doing simulations of this network, and well, and how? Okay, see, I read that you did 1 million model simulations, meaning that 1 million different choices of parameters.
Yeah,
who had selected within sort of some predefined range according to literature and
exactly.
And then how long biological time did you do for each of these simulations?
Yeah, so each simulation is about real time. So if you run a minute of simulation, so I ran, I think, 200 seconds per simulation in the paper. That takes about a minute or 2 minutes. So they're pseudo real time.
Okay.
And then you can parallelize these simulations basically one per CPU. So I ran it on our compute cluster parallelized across 128 CPUs. And this is using a very nice package called Brian 2. Where basically somebody else is taking care of all of these problems on the programming side.
Yeah.
But you can run hundreds of these simulations in parallel. So I think I ran, 500,000 or 1 million over Christmas break. Basically, I pressed go, went on holiday, and then came back. Here's 500,000 simulations.
Oh, very good. Well, that's always a good feeling to have your computer work for you when you're on vacation.
Right.
That's, so, and then of course sometimes you come back and you've done something wrong, so it's all
Right.
useless. But maybe you were better prepared. So okay, so and also say that then you had this 1 million model simulations, and then about one quarter of them,
Yeah.
260,000, were what you call valid.
Yeah.
So what does that mean? I mean, they were.
Yeah. So this was like the problem I was telling you before. So this 1 million random parameter sets were randomly sampled, right?
Mm,
From like a uniform prior distribution. And
mm,
so if you just randomly sample models like this, most of them actually don't do anything or they explode. So they're just like
okay.
completely synchronized.
So.
And neither of those regimes are very interesting, uh, both scientifically and
Huh,
then on the machine learning side, because you can't really learn anything from that data. Right? So
yeah.
there was this pre-selection step of, let's see if the activity they produce fall within some reasonable range, like firing rate and roughly, yeah, reasonable frequency spectrum and population covariances, etc.
Yeah. And one quarter of your simulations were okay, or at least
They
sort
were
of
okay,
reasonable.
yeah,
Yeah,
yeah.
that's interesting. Or it could have been much smaller
It could have been
fraction.
much less, right? I was also kind of surprised that it was, yeah, exactly.
Yeah. Hmm. Cool. And then so then you have this 1 million or 260,000, 60,000 simulations where you then compute. You have these 260,000 different models in some sense, which then have different simulated data, and then you want to use this to in the next step, right?
Hmm.
It's all this like simulate what's called simulation based inference, which I guess
Exactly.
SBI,
Yeah,
that was been developed in the Maca lab.
yeah,
So what was so what is this all about?
yeah. So at a high level, simulation-based inference is, I guess, a relatively recent nomenclature. Essentially, you want to use machine learning, so the type of deep neural networks, to fit parameters of a mechanistic model, in our case, the spiking network model, to data, right? So the machine learning is helping you do work. And how it does that is by learning the machine learning models learn from simulations, the mapping between the parameters they're interested in to the activity. So, and this methodology is really, yeah, it's strongly driven by Yakov's lab, but also there's many different communities that use this type of methodology, like in physics, to do gravitational wave inference, all kinds of, on a small scale as well in molecular biology. So that's the umbrella of simulation-based inference. And
Mm,
then, and the lab had a very nice package also called SBI simulation-based inference in Python. And this made a lot of these workflows basically much easier to deal with. And
mhm,
then sort of AutoMIND, where you call it, or what we call it in the paper, is essentially the combination of this SBI workflow, specifically using neural networks, with the specific spiking neural network architecture in the context of neuroscience, right?
mm,
Um, but yes, the logic is if you train a machine learning model to understand how a simulator goes from biological parameters to neuroactivity, then perhaps you can then give it real neuroactivity recordings and
mm.
ask to give you the answers back. So what parameters would have been likely to generate these
Yeah,
recordings?
that's sort of like the inverse problem, right?
Exactly.
Opposite
And
problem.
that's what we call it inverse model. Yeah.
Yeah. Mm. And so, what kind of architecture is used for this? Is it
Yeah.
what kind of machine learning architecture?
So in the paper, I end up calling it deep generative model, but the specific architecture that's used to do the inverse mapping is called normalizing flow.
Mm,
So it's a probabilistic machine learning model that gives you a distribution of answers. Um, how you can think about how it works is essentially is it starts from a Gaussian distribution, and the machine learning bits learns how to squish this Gaussian distribution in high dimensions
mm,
through multiple steps of the flow. into a more complicated non-Gaussian distribution that would capture your data well, right? And so the job of the neural network is to say, given a piece of recording, and that's the conditioning part, given a piece of recording, I want to know the transformation that takes from the Gaussian to this complicated distribution that would give you valid samples to reproduce this recording that you asked it to.
Yeah. Cool. And this is sort of like this deep generative models. That's a whole subfield of machine learning.
Exactly. So this falls very much within the realm of deep generative models. So for example, diffusion models, if you're familiar with how you make the nice pictures based on your text description, it's the exact same principle, right? It's conditioning on a sentence, which is text, and it learns how to transform. So diffusion models for images essentially learns how to transform these blurry pixelated white noise type of pictures into a coherent sensible picture based on the prompts that you give it.
Yeah. Cool. So, okay. So then you have this AutoMIND, which is sort of then this essentially this combination of this network model, spiking network model, and then this deep generative model on top of that, which together give you this system so that you can put in experimental data and get sort of not only a single, or you can get a whole distribution, or you get the whole whatever called the invariant sloppiness structure.
Yeah, you get a distribution of parameter combinations or valid models, and then if you analyze how these models are populated in the parameter space, then you observe things like structure in this distribution, basically. So it's not so what you can imagine the naive hypothesis would be you get a 28-dimensional Gaussian back centered at some point, which is the mean of the valid region, but moving around in sort of in a random fashion around this Gaussian gives you valid parameters back. But what we see is that's not the case. Very often you have non-trivial correlations between parameters. So you can find two parameters, for example, that are linearly correlated. So they compensate for each other. And this structure is what we call the invariance.
Cool. So then to the first example, I think you applied this you follow this development of this human brain organoids and measured spikes from this over a period of 40 weeks or something. So first, what's an organoid?
Yeah. So this was magical stuff, which I learned about during my PhD. And this data was actually collected in Alison Moultrie's lab at UCSD. This was in collaboration still with Brad when I was doing my PhD. And an organoid basically is an artificially engineered piece of tissue. And a brain organoid, or a stem cell derived brain organoid, is when you take, so typically what happens is if you take skin cells from a person, which is already a differentiated piece of tissue or cells, you can turn them back into stem cells by, dumping on them a soup of magical drugs.
Exactly. Molecular magic.
Exactly, molecular magic. And, somebody got the Nobel Prize for this.
Yeah.
And then quite recently, actually. And then if you take those stem cells and if you dump a different soup of magical drugs, they can become various kinds of tissues, in this case, brain tissue. And specifically these 3D brain organoids, if you keep them in a condition where they're, kind of floating and moving along over the course of many weeks or months, they develop a 3D structure. Yeah, so that's what a brain organoid is.
And how big are they typically? They are sort of like...
they're typically on the scale of like 1 millimeter cube, up to from on the order of 100,000 to a million or 10 million cells, in that range.
Huh. And then you put them on this. How do you measure spikes from them? Obviously. And
Yeah, so then
then,
if you take the organoids and you put them on a multi-electrode array, as you would in regular 2D culture as well, you measure activity over time. So,
mm,
and in the original study in 2019, where what we reported basically is if you keep them for a very long time over the course of, I think like 9, 10 months, basically, you see pretty interesting evolution of their network patterns, the firing patterns of these neurons in the network.
mm. So yeah, what kind of features did you focus on?
Yeah, so at the very beginning, once you play them, they start to kind of get comfortable. You see very sparse single neuron activity, but very quickly they get into this like synchronous state where essentially the whole plate fires at the same time. And then they pause for a very, very long time, up to a minute or 90 seconds, and then again. And it's a very stereotypical pattern with very stable pauses between these network synchronous events, as well as what the shapes of the events look like. And as they get more and more mature, the pauses between these network bursts become shorter and more
Mhm.
variable. So that was the aspect that we try to capture with this network model is, so over the course of the 40 weeks, if you look at, for example, one recording per month, let's say, you
Mm,
see that, yeah, the network bursts become shorter and more variable. And so those are the patterns that we try to reproduce in the spiking neural networks.
mm. So
At least
that's
that's what I wanted to start doing when I was still a PhD student, and then ran into all these problems with fitting networks.
mm. Exactly. So you try to develop by hand sort of
Yeah,
networks that could
exactly.
sort of give this
Right,
burst duration and the variability of bursts and yeah, yeah, exactly mean interval between bursts and all kinds of things.
right.
So now with this AutoMIND, you could actually make some progress and
Yeah,
get results, right? So then you could actually
exactly.
so then for each of these every 4 week you got a new sort of like an ensemble of models,
Hmm.
and then you could sort of explore the parameters of these ensembles and how
Right,
this ensemble varied over this 40-week period.
right.
Yeah.
Yeah, so the scientific question here is basically we observe the electrical patterns change over time, right, over the course of 40 weeks. And there are still many aspects of the network that we want to understand but can't directly measure or is very inconvenient to measure, which are these properties which we then try to model to put as free parameters in the model. Because at the very beginning, my question was simply, well, if their activity changes over time, then you could probably see a parameter that tracks similarly this evolution, right? So for example, I don't know, like the number of excitatory versus inhibitory cells, that's one hypothesis. And
Mm,
so the expectation I had at least was if you fed a recording using this type of methodology, whether by hand or using machine learning like we do with AutoMIND, you will
mm,
get essentially a mean change of the parameter over time, right?
mhm,
But then having done this exercise, uh, several years later, what we do find is actually, first of all, there's a big range of the distributions are quite broad for most of these parameters,
mm,
um, and also that you have these structured or correlation structures within the parameter space across different parameters, and then that both the means and the structure of correlations changes over the course of development, right? So
mm,
that's quite interesting. So that means there is not a single, or there were some parameters that did evolve in this expected way, where you can clearly see that the means are kind of changing and the distributions were roughly similarly spread. But a lot more parameters behave in very weird fashions, right? Like they were just broad all the time, or they start off very broad and collapse. And then the
mm.
relationship between parameters also change over time.
Yeah. Ah. So exactly. So the thing is, which is one, I guess, key take-home message is that you have maybe like two parameters which pair up, and if one goes up, the other one can maybe go
Yeah.
down, and to end up with the same function. And that
Right.
means that that sort of then it's obviously you expect sort of just a large variability, large span, large range of each of these individual parameters. So you
Hmm.
really have to look at the correlation structure. But
Right.
one thing is, I guess, look when I looked at the paper, that this leak conductance of the neurons that increased with time.
Hmm.
So maybe is that sort of like there are more and more maybe channels being inserted into the membrane, maybe to increase the leak conduction as this organoid develops. Is that sort of a possible way?
I honestly don't have a great idea of what the what would be the actual mechanistic explanation to these changes in the model parameters, right? Um, I think leak conductance and capacitance, they're fairly simple physical explanations for them. But I also don't know if those make sense. But then if you get into like sodium potassium channels, especially the voltage activated ones, first of all, the model itself is phenomenological in the sense that, for example, there's this adaptation parameter that tries to capture all these different things at the slower time scale. So that's still a fundamental limitation of the model that I ended up using. So if you switch the single neurons out for a more realistic model like the Hodgkin-Oxley neuron, for example, then you can put in different like physical ion channels that that you can exactly map to the ion channels in the real neuron. Then you can start to play these games where I can manipulate or at least track exactly what biologically it corresponds to.
Wow. So you think that for this, your choice of model made that of rather simple neural models made that maybe a bit difficult for to do this kind of interpretation in terms of direct biological channel or channel, yeah, membrane
Yeah,
mechanisms.
it depends on the resolution or the like how mechanistic you want to get to the actual biology, right? There were some parameters that did map to physical things, but then there were other things like what I mentioned, the adaptation time constant, for example, that's just totally phenomenological, where
Uh,
multiple things mix together.
what is the adaptation?
And
That has to do with the ADEX model, or
yeah, exactly. It's the
yeah,
adaptation equation in the ADX model where
well,
after a spike happens, or there's the stop threshold part and then there's the active part,
mm,
where if a spike happens, then there's spike frequency adaptation, then the next
mm,
spike kind of adjusts accordingly. So that process has a certain time scale as well. We try to fit that and usually has a longer time scale. Um, but yeah, so you're always playing this game of how complicated am I going to make my biophysical model to how tractable is it going to be and how useful it would be to distill some abstract principles of how a network works, right? Because at the very extreme end, we can have something like, I think you had the Python Diceman model on the podcast a couple months ago, right? And then
yeah,
even more crazy is the blue brain model, where you have a very morphologically detailed
uh, yeah,
model of the whole cortex
mm,
or a colony of the cortex up
mm,
to now we have single neuron point models or even like mean field descriptions like neural mass models where they have two nodes that basically capture hundreds of neurons
uh,
activity. So you're always trying to play this game of how biophysical am I going to get to the explanation versus how kind of convenient and how much abstract things you learn out of the model.
but how? I mean, say if you had sort of replaced your models, your neural models, with some kind of Huxley style models, and then instead of have like 28 free parameters, you had 50. I mean, so what would be the problem with that? Mainly is sort of to run all this forward models. Is that would be the what will stop you from doing that? Or is
Yeah, the main
yeah,
problem is computational complexity. So if you open up the parameter space, then you get exponentially number of combinations, right?
yeah.
Um, and so, first of all, sampling the space for valid model configurations will be more difficult,
Mhm.
which means you have fewer valid simulations to train the machine learning model with. And then on the flip side, when you're trying to do inference, you're essentially approximating a much higher dimensional probability distribution, and that is difficult. And then, yeah, and in general, it's this search problem in high dimensional space. And the more parameters you're allowing, the more difficult it gets.
Oh. But say I just was like, if thinking out loud, if you had then this computer power available, both for doing this forward modeling and making
Yeah,
this inference model, uh, would it be also be limitations if you have a fixed amount of experimental data? There must be some kind of limitation to how much you can how much you can extract, how much you can learn about the parameters from a finite amount of experimental data,
yeah,
I would guess.
yeah. Totally. I think this then gets back to the technical point of
Yeah.
like underdeterminedness, basically. Like if you have a
Yeah.
system of equations and you have
Mm.
much more degrees of freedom than you have the ability to constrain them,
Yeah.
then you get a space of solutions that would be all valid. And
Yeah.
if you add additional observations or external data,
Mm.
that's sort of
But one
orthogonal to
thing
that.
I guess, because I think here as a summary statistics, you had like the mean the interburst interval and the
Yeah,
variation and the burst duration. So you had three summary statistics,
yeah,
or but you could have been more ambitious to have, instead of three, some other kind of
yeah,
higher
exactly.
correlation structure, and that could probably then constrain it more than in
More
principle. Yeah.
exactly. And
Uh,
um, that was the point of the other analysis with the
mm,
NeuroPixels data set, is say if we use the full frequency spectrum, which in that case was 1,000
mm,
dimensional, I think, right?
yeah.
So at 1,000 frequency bins, which is a very high dimensional observation space, would that constrain the models better, and maybe get like very few parameter combinations that can explain it? And even in that case, you still see, first of all, fairly broad distributions, and you also observe these correlation structures across parameters.
Yeah. Maybe it's just, yeah. So now so now we can focus on that. So you did had this neuropixel recordings from mice, and where you sort of both measured spikes in, I guess, was like some hippocampal areas and some visual areas, and then, as I said, you used a much richer, yeah, that were more ambitious in the sense of what you wanted to the
Right.
model to be able to predict, the firing rates and a normalized like power spectral density
Mm-hmm.
of the spikes. So okay, so what was the outcome of that? How much did the, maybe difficult to compare, but how much did the, yeah? So you had more data then, or so you used it because you used the same forward model. You used the same,
Yeah.
yeah. So you used the same like 250,000, whatever, 60,000
Exactly. Yeah.
simulations, and then but you had a different, obviously then different like summary features that you wanted to
Yeah.
predict.
Right.
Okay, and then you had more complicated, more higher dimensional summary features. So
Mm-hmm.
how did that go compared to the organoid thing?
Yeah. So in this case, the simulator model and all the simulations that you use to train the networks are the same. But at the very end, we basically, you have to compute, yeah, these summary statistics differently. So instead of the three bursting features, now it's this 1,000 dimensional spectrum. And that is the thing that the machine learning model learns to map the association of those things to the parameter sets, right? Because at inference time, or when you have a real piece of data, what I was interested in capturing are these full frequency profiles of the population firing rate in these different areas.
Hmm.
How did that go? It was pretty much business as usual. Essentially, you just have to retrain the machine learning model to account for these different features.
Hmm.
Um, and then the rest of the exercise is the same. What I can tell you is that uh, the answer that gives you, when you give it a real piece of data, it gives you a bunch of parameter in this distribution, right? There are fewer of those are valid. So what I do in this, there's this like post hoc selection step of, if I ask the deep general model for, let's say, 1,000 combinations, not all of them will be able to reproduce the observation, even though in theory it should be able to. But so practically, there's this post hoc selection step where I simulate them and then essentially keep the ones that uh, do behave similarly as the observation under some metric. In this case, like the mean squared error between the observed and the simulated spectrum.
Mhm.
And then I do all the analyses on
So I'm a bit
the
confused. So you
valid
had this
ones.
260,000 that you called valid and you used for the organoid project, but then you did something
Yeah.
in addition.
No, and those were still the same. So that
Yeah.
training set was always the same.
Okay.
So you use that big training set to train the machine learning model, whether you use the bursting feature or the power
Okay.
spectrum
Mm.
in both those scenarios, right?
Mm.
But when you at like inference time, when you have the real piece of experimental data
Uh,
and you input that into the machine learning model and you ask it, I want to get 1,000 parameter
yeah.
configurations, right?
Okay.
So what I was telling you is that not all 1,000 of those parameters that the machine learning model answers for you will give you the exact same observation as you asked it to,
Ah. So
for a variety of reasons.
okay.
But essentially, it requires so then it wouldn't make sense to analyze those models as if they were a
Mm.
good fit of your data.
Yeah.
So practically, there's
Mm.
this
I
post-hoc
see.
selection step. I just keep the models that end up looking
Yeah.
like the data.
I see. So it's like an extra sanity check in some sense.
Exactly. Yeah.
Yeah.
Yeah. Like a
That you
filtering
sort
step.
of,
And I mean, that's one thing that you should always be doing if you're trying to fit a model to data, right? It's like, yeah,
yeah.
your model actually ends up looking like your data.
Yeah. Well, that's true. So I'm just sort of, I mean, uh, in like when you people talk about like foundation models. We had a podcast with Andreas Tolles about foundation
Yeah.
models, and then it turns out that you can sort of if you have you can sort of do this pre-training. So I just wonder if this if you had this network, or maybe that doesn't make sense. This I'm just thinking if you had this deep generative network that was
Yeah.
trained for the organoids, if some of the training of that could be a better starting point for this.
Yeah, that makes perfect sense, actually. You can think about this whole exercise as training a foundation model with simulated data instead of real
Mm.
data, right?
Yeah.
So Andreas' stuff and this whole foundation model paradigm essentially is I have a massive amount of real data and I use some objectives that is not supervised learning, but unsupervised or self-supervised that essentially just tries to learn the patterns just from the recordings itself. And that massages the machine learning model in a way where it's kind of familiarized with what real neural data look like.
Mm.
But so you can totally replace the real neural data with simulated or synthetic data and kind of massage your machine learning model to in a very similar way, right? But that requires your simulator or your mechanistic model to produce things that roughly look like your real data. And if that's true, you can then play this game basically of replacing real data with synthetic data. And this is a thing that actually people do in the machine learning literature. There's different names for it. Some call it prior fitted networks. Essentially, you're training these big foundation models with purely synthetic data generated under some statistical model. If it's a time series, you use like Gaussian processes or something, or Brownian motion, various combinations of things. And then in the end, you can deploy that model to and ask questions about real data, basically.
Mm. Mm. Cool. Yeah. So you could even think this could even become like a community effort in some sense.
Yeah, totally.
Mm.
You could try to develop, and I think that's kind of the goal of mechanistic modeling in some sense, is to have a model where you can produce data that looks like your observation, right? Like if you look at, physics, chemistry, that's
Mm.
what models are for. Obviously, then you want to understand or you want to be able to dissect the model for principles behind those observations. But first of all, you require your model to behave in a way similar to what you observe in the real world. And so there's this cyclical nature to this where you want to develop better and better and more realistic models, and then at the same time extract principles from them. And so if you have really good mechanistic models, you can supplement your real data with simulations from there to train
Mm.
a machine learning model. and then learn better parameter distributions, in this case, biophysical parameters, to make better simulations. I think that's a healthy and productive feedback cycle.
Mm. So, I mean, one thing to test, I guess, these models on when you have these ensemble models is to drive with like stimulated with new kind of stimuli, right? I mean, or maybe increase the input or something to see and to check whether the predictions from a model corresponds to a
Right.
perturbation from an experiment. You did that. I think you mentioned in your paper that you did this for the
Yeah.
organoids.
Yeah, we did it for both the organoids and the mice, or yeah, the mouse models. For the organoids, what we did is there was a separate experimental recording where they blocked GABAergic synapses. And so if you do the same manipulation in the models, which were constrained to not that recording, but the resting state recording, and if you block their GABA activity or GABAergic synapses, will it reproduce the other recording, right? And then in the mouse, what we did was just increase the input into the excitatory neurons, because
Okay.
in these areas, like hippocampal areas, you have these gamma oscillations, and there's fairly well-known experimental and theoretical work saying if you increase drive, certain things happen to the oscillations.
Mm. And how did that go? Did the models that you sort of typically did the models that were fitted to the baseline? Do they typically predict what happened during the perturbation?
Yeah, yes and no. It differed between different cases.
Mm.
So I think in the example I put in the paper for the CA1 recording, most of the like baseline constrained models, when you increase input by 50% into the excitatory neurons, you do get the expected behavior of both increased frequency and amplitude of the gamma oscillations, right?
Mm.
But how much it changed by varied between the 200 models in this ensemble. In the organoid case, when you block GABA, or in the model, you just set the inhibitory synapses to zero, right? When you do that, not all of the models end up producing activity that's like the perturbation recording. Um, and we have some hypothesis as to why, but yeah, not all models do the thing, end up reproducing a perturbation experiment.
But I guess I'm just thinking if you had sort of instead of just training your model, say, to the baseline data, but train the model to, or yeah, train the models to predict both the baseline data and the perturbed data, then
Mm-hmm.
probably you would have only ended up with these successful models.
I would tend to think so as well, or at least your likelihood of success becomes higher. But then you're sort of, in that sense, cheating by having given the answer
Yeah,
ahead
no,
of
that's
time, right?
true. But I'm just more thinking of if you have, I mean, I think in terms of systems identification, then of any
Yeah,
system, then you have baseline and different kinds of stimulation. You have like a
right,
really
right.
rich data set, and then of course you have to do the same thing in a model. And
Yeah,
I just wonder if you get better, if you have a larger, richer data set, then obviously that was a larger, more complicated model simulation. But
right.
then you would end up with better parameter sets because they are sort of tuned to deal with more situations.
Yeah. So that is one thing that I didn't do, but would have been very exciting. I think, I mean, I will end up doing this, is that let's just take the input case, right? So
Mm.
right now, to the model, I only give it Poisson drive, and the free parameter is basically how strong the Poisson drive is. So it's
Mm.
random Poisson spikes. Um, you can produce simulations with under a variety of input regimes, and then observe obviously different activity. And then you have a much, like I said, much richer data set to both train a machine learning network on. But then on the experimental side, you can then perturb or give the same kinds of input, different inputs to the mouse brain or to the organoids or whatever. And then from a system identification perspective, probably be able to narrow down to a much smaller set of valid parameters.
So, yeah, cool. So it sort of is a game where you can sort of decide what to train the model on, and the number of
Yeah,
parameters you choose. I mean, it's like the whole, it's like, well, it's more like a program than a project, right?
yeah,
Yeah, it's sort of
exactly.
like so many ways you can try out different things, but it's certainly, as I remember, I remember when I went to, I think maybe I told this story before on the podcast, probably have, but it's like when I went my first meeting in neuroscience, and it was actually John Hopfield giving a talk. This was a meeting in Sweden in the late 90s, and he said, well, in neuroscience we have to learn to compute without knowing all the numbers, and that's very difficult.
Mm.
And in some sense, this is a way to compute without all the numbers, right? That's sort of what this whole approach with that you're doing is, right? It's a way to well, you don't know all the parameters, so you have to sort of
No, no, you don't.
yeah,
And
and then you
and
have to make these ensembles and sloppiness spaces and everything, and think about it.
right,
Yeah.
right.
Hmm.
Yeah, exactly. Like so my goal again at the very beginning was just to identify these biophysical properties in these models because they often mean things, right? In a disease context, for example, if you have a slightly off neuronal property or synaptic property, you end up with neurological or developmental conditions. And so it'd be very useful to be able to take somebody's recording, whether it's like EEG or intracranial ECOG or whatever, feed it into such a model, whether it's machine learning model or mechanistic model, and have it be able to tell you these are the range of values for this person's like sodium conductance or whatever,
Yeah.
right?
Yeah.
And that would be a very helpful sort of clinical tool
Absolutely.
as well. So that was the dream, but then when we end up doing this, it would end up being a mess of degeneracy, and that's kind of like the takeaway here is there's a lot of degeneracy, and
Mm.
there's both the technical but sort of uninteresting degeneracy in the systems identification sense, and then there's the interesting and real degeneracy from the biological robustness perspective. Right? So then having more and more data essentially pushes out, it removes the possibility of technical degeneracy, and the
Mm.
thing you end up in the end, if
Ah.
there is any degeneracy at all, ideally maps exactly to the degeneracy in your real system. And that's
Mm.
now something that I'm very interested in trying to identify in these network models.
Yeah, exactly. Of course, then if you end up with well, if they end up with this biological degeneracy, then of course that's just how it is, right? And maybe then if you want to,
Yeah.
you're still, in some sense, if a person is still sick, right? Then there's something even in this biological degeneracy not able to deal with it, right? So you just have
Yeah.
to,
So, and
you
then
have
when
to,
you get to that space of literature, there's all kinds of interesting theories. There's someone here, Peter Jedicke, and also I think across Germany. So Susanne Schreiber had a similar paper on this, essentially on the space of degeneracy and what that tells you. So one theory, this kind of gets back to your point of the aging thing again. Is like, but like
small,
under some disease condition,
if it's really small, you're close to death.
yeah, basically, like if the space of your valid solution somehow is smaller and smaller and orthogonal to the directions of perturbations that you face, then you get into a crappy state, right? And then if you think about in like real life, your brain and the organism as a whole has multiple objective functions to satisfy, right? There's metabolic concerns, but also sensory coding, fidelity, and all kinds of things. So it's like a multi-objective optimization game where you can't really satisfy any of them, but your degeneracy, your degenerate solution space has to somehow be able to move freely within that space
Mm.
to account for all these different things. So you get into very interesting theoretical considerations of why there is degeneracy and what would be like an optimal degenerate set of
Mm.
solutions.
Mm. Yeah. So, I mean, now one thing you did discuss in your paper, and of course, which is of interest to us, is sort of the how this AutoMIND can be reused because you obviously put in a
Hmm.
lot of work and there's a lot of tools acting together. And you've
Yeah,
also done this Christmas simulations, which
right.
we can if we can reuse them, that's a good thing for the community, because that
Yeah.
sort of is. I mean, you can sort of so how can you reuse AutoMIND to other experimental data or other? I mean, you other summary feature. You can sort of we can already take advantage of your sort of forward modeling of this whatever
Yeah.
260,000 models, and then use them with new experimental data and maybe new
Yeah.
sort of like summary features.
Yeah, so there are a few ways of reusing this here, right? The most basic way is if you just have new data, but you are interested in the same set of summary features, you can directly take the trained machine learning model, which I uploaded into like a big repository somewhere. You can just take that model, run it locally, give it your observations. So let's say if you record it from instead of the mouse hippocampus, I
Mm.
don't know, like the monkey parietal cortex or something, right? And but if you're still interested in producing the same power spectral kind of
Yeah.
observations, you can just directly take that array.
So then we use the same summary features as you did.
Exactly. And
Mm.
then you already get the same parameters, or you get the parameters for the same model out.
Oh, but I mean, yeah, cool.
And I've already sort of worked with some people that had started using it in this way with their in vitro recordings, right? So they had also either 2D or 3D organoid recordings where they want to reproduce the bursting features. So you can do it like that. The second way to reuse this is to retrain the machine learning model by using the summary statistics that you're interested in. So
Mm.
we have uploaded sort of, you can access the raw data. So because of simulation, we have access to everything, right? So we have the
Mm.
exact spike times for all these neurons in the network that we recorded from, and you can recompute whatever features you want from those spike trains.
Mm.
So you can then redo that analysis in the same way you would analyze your own data. And then you would take that simulation set of a different set of summary features from that simulation,
Mm.
retrain a machine learning network, and now you've done this exercise yourself. You have a different trained deep generative model, and now you
Mm.
can produce, or you can estimate parameters of the same model using your observations and summary statistics. But you're always constrained in this case to the simulator model that I used.
Yeah, exactly. So then, uh, so how do you think about that? Because now you use this, uh, because your network model was used separately for this. I think for now for the second project, NeuroPixel. So you had like six different areas,
Mm-hmm.
CA1, CA3, and so on. And you found a network model, and you fitted models individually for these six areas, right?
No, the machine learning model is fitted once. You just
Huh.
have to recompute the power spectra, or just compute the power spectra from the 260,000 simulations,
Yeah.
and then you fit the machine learning model once.
Yeah. But I mean, but you get sort of you get six different results, right? Because
Yes, exactly. So
you
I
get
have
six different.
six experimental observations or recordings
Mhm.
from the six different areas, and
Yeah.
then I, enter them independently and get
Mhm.
six different sets of
Oh.
parameter estimates.
So what is this? I mean, so this obviously, if you go into the details of these different these six areas, hippocampal cortical areas,
Yeah,
they are quite different, right? So,
yeah,
so in some sense, it's like this one size fits all circuit
right,
model that you sort of.
right.
But maybe that's. I just wonder how do you think about that? Maybe that sort of is that you how you yeah. It sort of is like this. Obviously, this rich dynamical system that is able
Yeah,
to, but it doesn't necessarily have to be too close to biology.
yeah. So off the bat, what I can tell you is obviously the real visual cortex and hippocampus don't really look like
No,
each other, right? I mean,
no.
there's differences in layers, differences in the cellular architecture. There's millions of differences. So,
Mm,
what is the point of using the same model to explain both of these different areas, right?
mm,
Um, one of the questions is just are you able to do that, right?
Yeah.
If you gave a spiking neural network enough flexibility in these free parameters, obviously the model was designed in a sort of general way, or it was in consideration to be generic, such that it could kind of roughly look like many different brain areas. And the free parameters themselves kind of determine what it looks like, depending on the activity that you constrain it by. But so yeah, so the question I had was, if you have a flexible enough spiking neural network, do you really need to have the sort of biologically grounded details that we know about these areas that I didn't put into the model to reproduce those activity? Um, and for the specific summary statistic that I was looking at, I guess the answer is no.
Yeah. But I guess there are like this if there's sort of like this two goals, which are, I mean, one goal is to be able to make something that reproduces the recordings and maybe are able to predict the responses to all to new stimuli. And the other one is sort of being something which is close to biology, so that you can actually
Yeah,
learn about the parameters and maybe come up with a suggestion for a drug or something. And these two,
right.
that's like two slightly different goals, or
Yeah,
quite different goals, right? So I guess you have to try to to navigate yourself, well, navigate yourself on that sort of axis according to what you're most interested in.
right. It really it's a function of your question, I think. And if you know, and we do know again that there are differences between these brain areas, right? And obviously, then again, a mouse brain does not look like is nothing like an organoid.
Mm.
Nevertheless, I use the same model to reproduce activity from all these recordings. So yeah, I wouldn't be able to say like the things that we learn out of this model should map exactly onto those brain regions, as like we learned about that brain area from this, right? But I think that is the goal, is to have then tailored models to different brain regions,
Mm.
putting in as much of the biological details that you think is necessary, and then for the other parameters that you're not so certain about or you just don't have measurements for, fit them to data.
You mentioned
Um,
that, absolutely. So and then, so this thing of designing new circuit models or new models tailored for different areas or different problems, or, and then you mentioned that you, I think you worked something on misspecification aware algorithms, which is that
yeah.
the saying that, oh, that you can actually have some algorithm saying that this was a stupid model to suggest for this particular data.
Right. Exactly. So there's this general problem called misspecification, which is that your simulator model or your mechanistic model is not really a good description of your like underlying physical system, right? And if that happens, then the simulator will never produce data that looks like your observations from the system.
Mm. Mm. Huh.
So this is kind of actually the complement of the thing that we were just talking about, because we were talking about a simulator that you know is wrong but produces activity that still kind of looks like your real observations. In the second case, it's if you have a simulator that you know is wrong and produces stuff that looks nothing like your observations, what do you do in that case, right? Or you might even think the simulator actually is fine. The model roughly looks like my underlying biological system, but it just doesn't have some sort of nuanced ingredients to reproduce the activities. What do you do in that case? Um, so then you can design inference algorithms that's sensitive to this type of misspecification, or first of all, uh, quantify or detect when such misspecification happens, and then come up with methods to basically close the gaps and just say, this is the model I'm happy with, and let me try to find a configuration that's as close to my real data as possible.
Hmm. Hmm. Cool. So, I mean, a little bit about your. I'm not quite sure what you're going to focus on in your new job, but I think if you sort of wanted to pursue this kind of move, I think this is really important and not easy, but very certainly the way important avenue to explore. So what is the, if you want to sort of to, about the future developments of this, because I think, I mean, there's some parameters that really vary with time. I mean, for example, I mean, during development, for example,
Yeah.
the leak conductance, because you get more channel densities vary with time, because that has to do
Hmm.
with sort of how they are sort of transcribed by in the, well, inside the cell. So that is something that can vary over time. Uh,
Right.
but then there are other ways to describe this sort of. I mean, you can sort of model. You can have the models describing the transcription, where instead of getting
Mm.
like you have like static parameters maybe for
Right.
this transcription rules, and so that you end up with a set of static parameters, and parameter doesn't change over time,
Right.
instead of like dynamic variables. Is that sort of a that is that something you dream of, or?
Yeah. That is something I dream of, and I'm going to give you a very specific example of people already been doing this. I think you had Tim Fogels on a couple months ago, or like maybe a year ago. They were doing this type of exercise, but with synaptic plasticity rules, right?
Mhm.
So that is pretty much the complement of what I was doing, and this kind of closer to what you were saying, because the then the network is able to evolve within like the course of a
Mhm.
single simulation, right? Just by
Yeah.
these like time dependent plasticity rules. So in that process, the circuit then settles on. They were trying to find these like asynchronous irregular states. So what learning rules facilitate that? So that pretty much goes along the lines of what you're
Mhm.
talking about.
Absolutely,
So
exactly.
similar, you can think about like RNA expression into synaptic receptors or ion channels. It's very complicated. It's all very complicated, right? Because you have multiple resolutions of biology here, or multiple scales of biology, from molecular stuff up to, subcellular, cellular, up to networks. All of these things are dynamic. I think none of them are really constant in any sense, right? Like we have in our model static network connectivity weights that don't change. But
Mhm.
obviously, even within very short timescales, these synapses do change through facilitation, depression, etc. So all of those variables in the end are dynamic. You have to make the choice of essentially make a timescale separation of saying these things I'm going to say as fixed and constant, and these things I'm allowed to vary, like brain activity or spiking
Mhm.
activity.
Yeah.
Hard to answer that question for you. All of this is interesting. I think progress really depends on what is tractable at a given time, both in the modeling side and also the machine learning side. Obviously, you have to be able to run these simulations at a reasonable time frame, right?
Mhm. So I mean,
Um,
actually, so this is very much sort of compute limited. What you can do,
yeah,
yeah, which is some sense a good thing, or a meaning that is something we can we can maybe get better computers, right? So it's like
yeah.
yeah. So it's not like
That's the
it's not a principal problem.
that's the optimistic
Yeah,
way of thinking about this problem. Is like
yeah,
at least within some time scale, like within a few years, Moore's law takes over. We have faster compute, more memory, and you can basically scale this up, doing nothing else different but scaling up to more simulations, more parameters. So you can always do that. And then there are, on the machine learning side, you can develop cleverer, basically network architectures and inference or learning
yeah.
algorithms to better capture these, for example, the degeneracy that exist, so you can sample the parameter space more efficiently.
Mm. But it's interesting because I think many people outside the field would think that you need bigger computers so that you can run bigger networks. But that's not what you really need is bigger computers so that you can run small, like one network really, really many times, right? That's
That's the
really the,
philosophy here, exactly. So you don't need a gigantic cluster to run a single blue brain simulation, for example,
yeah.
but you can instead run, hundreds of thousands of parallel
So it's isn't it so-called
simulations of
like
the same network.
embarrassingly simple parallelistic?
Exactly.
Yeah,
This
yeah, which is
specific network is embarrassingly
that's a good
simple
thing.
or parallelizable.
Huh. So and then you mentioned this thing about getting better like well machine learning models like deep generative models, I think you mentioned, I think you already spoke a little bit about it, but I didn't quite get it, how diffusion models can help.
Yeah.
So how is that? Or what is, I am not exact, what is a diffusion model in this
Yeah.
context?
So diffusion models in the machine learning context, probably the thing that people are most familiar with is stable diffusion, or I forget what the thing now like ChatGPT uses to generate image models, but basically these image models that creates nice realistic pictures. And how that works is if you think about a picture as a very high dimensional data vector where each pixel is basically one element in this vector, right? It's the exact same problem here of mapping or of trying to find samples in this high dimensional probability distribution. So the diffusion model in machine learning, how it works is it samples some Gaussian white noise in this high dimensional space and then tries to find a path into samples of your realistic probability distribution. In the image case, the probability space of all reasonable pictures, right? And then on top of that, there's the conditional part. The conditional diffusion model says, I want images of a specific description, right?
Okay.
So you can guide the diffusion trajectory into spaces of pictures that fits your description. So if you give it a text prompt saying, I need a picture of a brain that is half blue, half pink, and has a third eye or something, that specific text prompt vector will then guide the noise denoising process into a space of images that fits that prompt. So it's conceptually very similar to the normalizing flow. It has some properties that's advantageous and some disadvantages. Namely, it operates in higher dimensions, so you can get bigger parameter spaces, basically. But conceptually, it's very much similar.
So in the context here, you would sort of use it to end up with the models that are in this whatever, like sloppy invariant sloppy space.
Yeah. So the text prompt in that case, what I was trying to explain before was the text prompt in my case is the activity vector, right? So that's the power spectra or the bursting features. So
exactly.
when you ask the model, whether it's the diffusion model or a flow model, you just say, I want a distribution of parameters that will be consistent with this prompt of activity, right? And it transforms this Gaussian distribution into this data fitting posterior or conditional distribution.
Oh, very good. Yeah. Wow, it's like the ideas from all these cool ideas in from machine learning.
Yeah. In general, I think there's a lot of useful things that you can just taking machine learning or AI methods and help us do our regular computational neuroscience more efficiently or better. Never mind the sort of representational similarities within these networks, which is a different but also interesting sort of trajectory of research.
Yeah. So I think if you want something like one, we had had a podcast that was like a few months ago with Nicole Rust about sort of like the essentially about why it's like these elusive brain cures. Why has it been so difficult to find sort of going from the lab bench essentially to the bedside when it comes to curing mental ailments, mental ailments or neurological disorders, and I think one thing that and one her point was that it's like this idea of like single factors determining that you can fix like this like chain of dominoes. You have to think of it as a complex adaptive system, the brain as a complex adaptive system. And I guess this kind of approach really highlights this, that it's not this single factor mechanisms that makes the division between healthy and sick. That's not really how it is.
Yeah, exactly. I think translating that into the language of the sort of model that we've been talking about now is, so in my model, you have 28 parameters, free parameters, right? I think the classical view in this type of modeling work is that we can change one of these parameters, increase it, and something will happen. And often our hypotheses about how the brain is different between, let's say, wild type and condition A is that there's a single factor that is causal to this change.
Mm.
And and the last level we've been talking about is the degeneracy component is that very often you find that that's not the case. You can change this one parameter and the network output can remain essentially identical as long as there are wiggle room in the other parameter dimensions that are able to compensate for it, right?
Mm.
And
Mm.
the non-identifiability issue aside, if you believe that this degeneracy that we found is true of the real brain system, then of course, right? Like, and in that case, so the elusive cure is one perspective. The other perspective is how are we even functional like all the time? Because we constantly receive perturbation, all kinds of perturbation, right? Like, you went to a wedding this weekend, didn't sleep much, got drunk, and I wake up the next day and you're fine. So, and that's kind of like a miracle of our brain
Yeah.
and our like body in general, right? So,
Yeah.
and that's like a massive. I mean, depending on what you do at weddings, it could be a massive perturbation to your brain and
So
to your
that's
system as a whole. But the next day you wake up and
yeah,
you're totally
like
fine.
a
And so, how does that happen?
elusive miracle.
Exactly. So, if you think about it, it's actually miraculous that that our biological systems and our brain in particular is quite robust to all kinds of natural and self-induced, perturbations. And maybe degeneracy is the answer, maybe it's not. But what we do know is that there's a lot of sloppiness and stochasticity in biology, and maybe it gets used in a sort of advantageous way, such that different heterogeneous components can compensate for each other, such that you know these kind of single factor perturbations don't end up catastrophic all the time, right? We do have very good examples of that, like single nucleotide uh, perturbations, where if you removed or if you changed one gene in your genome, then something catastrophic happens. But I think
Mm.
for mostly our neurological and psychiatric disorders don't work like that.
Mm. Mm. Yeah. Okay. Well, it's been a very nice discussion. I learned a lot, and I really like this kind of work, and I think it's very important. So, do you have is there anything you want to say before we close off?
Um, I mean, there's tons of things I could talk to you about. I guess I just want to say thanks for having me on the podcast. I told you this before, but like a lot of the LFP modeling, the biophysical modeling of the LFP work was really instrumental for, especially the earlier stages of a PhD, but still even now, right? Because I think a really great goal to strive to both scientifically and for the translational and clinical sense is if you can take signal modalities that we have access to in the human. I
Mm.
mean, LFP ECOG is still invasive, but it's much less invasive than chronic single neuron recordings where things are much less stable, right? If you can have ECOG or even EEG and have a forward model of that biophysical process to understand what exactly is happening, and then invert that process such that given arbitrary signals from different people, you can make some estimates of their underlying biophysical system. I think that would be a really productive outcome for neuroscience, right?
Yeah.
Kind of
Well.
using these kind of computational and digital methods to peek inside the brain where you don't have access to measuring all of these,
Mm.
cellular things or transcriptions or whatever in the human brain, especially.
Mm.
So, you know, I guess just want to say like huge fan of your work this whole time, and it's going to
Thanks.
be
Well, thanks a lot.
very influential moving forward, especially as we move these type of spiking neural network models to generate LFP types of
Mm.
signals and then invert those. And I know you also have some work, on the meta modeling paper with Jan Eric, right? that does a very similar thing that tried to do the inverse model. So yeah, I think this is going to be cool in the next few
Yeah.
years.
Well, thanks. Well, I'm happy that you are now entering the field, or you've been there for a while, but at least you seem to have a you have a
Now I'm an
real
adult in the field.
yeah, you have a real job.
Yeah, exactly.
Yeah. That's good. Yeah.
Yeah,
So okay, well, thanks, Richard. It was very nice talking to you. So have a nice day.
same. Thank you.